Welcome everybody back to Paleo Talks. This week we have Dr. Greg McDonald back with us. Hey Greg.
Hey there.
And today he's going to talk with us about some of his research in Belize in the Cenote of Doom. And I can't wait to hear more about that name and the depths of this. It's an underwater cave and the bones are underwater. And I'm also working on some Belize projects. So this is something that Greg and I share, is these underwater passion and these cave passions for these Pleistocene faunas. Before we go further, I'll jump over to David and have him explain the show again and how it works.
Sure thing, same setup as usual. We're going to start off by having our guest presentation. So Greg, who we're happy to have back, is going to tell us about sloths. And then after about half an hour, about halfway through the program, we're going to open it up to questions from the audience. So at that time, we will remind you watching that to send us your questions. Leave them in the Facebook chat, and we'll be reading those out for the second half of the program. And as always, if for some reason you can't leave a question in the Facebook comments, head over to the Gray Fossil site on Twitter or Instagram, and I'll be keeping an eye there so we can make the second half of the program just Q&A with Greg.
All right, thank you. And we have Chris Widgood with us again.
Chris is here.
I think my voice is gone. I'm going to give it over to you, Greg.
All right. Sounds good.
I always do that when I talk about Chris. It's just.
All right. Well, fun to be back. Thank you, Blaine. And always fun to be able to share some of the projects I've been very fortunate to participate in. And of course, as everybody already knows, the common denominator is the fact that my motto is "Have sloth, will travel." So it's nice that people get a hold of me and let me know about new finds and provides opportunities for me to visit places. And of course, if you think about doing paleontology, Central America doesn't pop up all that often, but that's certainly an important place to be thinking about, especially with sloths, given they originate in South America and then have to come through Central America to get into North America and that. And, you know, we think of places like Guatemala, El Salvador. There's been a number of countries, Honduras, where Dave Webb, my professor at University of Florida, did research and sort of planted the idea in the back of my mind that this is a place you need to be thinking about. But one of the countries that does not show up often is Belize. And so we're just starting to scratch the surface with Blaine's work and my being able to work down there to show that there is a fossil record in Belize that needs to be looked at as well. So,
hey, Greg,
yeah.
Before you go any farther, I think we talked a little bit last time when you were on the show about your background and how you got into sloths. And if you could just tell the audience a little bit more about why you became a paleontologist, how you became a paleontologist. That's sort of something we're trying to do now.
Oh, sure. Well, when I was a kid, my mom was from Kansas. We lived either in California or in Utah, depending on the stage of my life. But my mom was the baby of the family, and my dad promised my grandma that he would make sure she got back to visit at least once a year. And so our family vacation was predicated on going from wherever we were living back to Kansas to see my grandmother and relatives. And then so we did that, but my dad always took a different route. So us kids got exposed to museums and national parks and all kinds of neat stuff on the way. And one of those stops was a place called Dinosaur National Monument. So you can guess what happened. I caught the dinosaur bug and got interested in dinosaurs. And we were living in Utah at the time, which of course is known for having a dinosaur or two, besides the monument. And of course, I'm sure my folks felt I would outgrow it, but I never did. I just like to say, well, I matured beyond dinosaurs and got into the Pleistocene. So basically, it was a family vacation, a stop at Dinosaur National Monument, seeing that wall of bones that fired up my imagination and got me interested in paleontology.
All right, thank you.
Okay. Well, obviously, people can pretty much figure out where I ripped off the title from or modified from. So we'll just call this not Indiana Jones, but Rhemaethium bones. Well, for those of you who have not thought much about the geography of Central America, this is Belize, which is on the coast. It's right next to Guatemala to the west and Mexico to the north. And the area that we're working is marked by the red star near the capital Belmopan. So it's pretty much in the center of the country. And on the right, a little close in there, we're working in a general region called the Yalbec Hills, and specifically at a site called Cara Blanca, which in Spanish means white face. Just to give you a sort of a feel of what the area looks like, that ridge that you're seeing in the top pictures, that is Cara Blanca, the white face. And the lower left-hand corner, you can see it's limestone. This whole area of Belize is basically on Cretaceous limestone, which of course extends up into the Yucatan Peninsula. And as we all know, limestone is a great place to look for caves. And of course, the caves then, and often, if you're lucky, have fossils as well. And basically, if you look at this ridge along there, it's along a fault line. So that ridge is basically an offset fault. And basically along that fault, they've identified, well, I think they started out with 25 and we're up to 26 or 27 of these cenotes that have formed along the fault line. So basically, you've got this whole series of cenotes that are probably connected by an underground cave system. And this is where we're doing our work. And for the reconnaissance trip I did down there, we hit 2 of those cenotes, because if you're carrying oxygen tanks and diving equipment and hacking your way through the jungle, it becomes a bit of a challenge. So we were focusing on cenotes that we could actually drive up to, because this area is actually being logged. It's on private land, which means nobody can get in unless you have permission from the landowners. But instead of wholesale removal, they're selectively logging trees. So it's here and there, they find a tree that they can log, and they leave most of the habitat intact. So you can see in the top picture, here's a cenote completely surrounded by dry rainforest. And at the bottom, you can see just, the forest comes right up to the cenote's edge. So why are we there? Do you just randomly say, hey, there's cenotes in the middle of the rainforest or dry forest in Belize, let's go diving? Well, that's definitely hit or miss. But the other area, of course, that Belize is known for in terms of research is interest in the Mayans. And this is a drone shot of a temple next to the cenote that was being excavated by Mayan archaeologists. And this temple is dedicated to the water deity Chac of the Mayans. And of course, we've often heard about the great droughts and the importance of water to the Mayan civilization and that. So Chac was a very important deity. And apparently, based on the studies of what they were finding here at the temple that were being led by Dr. Lisa Lucero out of the University of Illinois Urbana-Champaign, they noticed that there was a lot of items being brought as offerings that came from all over the country. And so the next question was, were these offerings also being thrown into the sinkhole, into the cenote, as offerings to Chac? So Lisa arranged for a couple of divers that run a diver that runs a dive shop on the coast, because most people go to Belize to dive, to look at the blue hole and to do the ocean diving, not to go inland into cenotes. And so they went in, they were looking around for Mayan artifacts, but noticed very large bones. And so that's kind of what got me pulled in, was the discovery of these bones by these divers looking for archaeological material. So why would I want to go to the ocean and look at coral reefs and stuff like that when there's water that's got bones in it? So I was very fortunate that when I was a graduate student at the University of Florida, the Florida Museum of Natural History offered a scientific diving class for anybody working at the museum. And I'd always wanted to learn how to dive. And of course, in Florida, you do a lot of diving in rivers and sinkholes there, looking for Pleistocene fossils and other material. So I got a lot of diving in when I was a graduate student at Florida working on my master's. And this is a skill that has served me ever since, such as providing an opportunity to follow up on this discovery here in Belize. And we worked with the dive folks from the coast that she had worked with before and put together a team to go down and to do a reconnaissance in a couple of these cenotes to just see how much bone was there and whether it was worth following up on. So this is not a drawing of our site, but it is illustrative of sort of what we're looking at, in that you have, of course, the cenote here with the opening to the surface. Water levels vary. They can often connect down to large underwater cave systems. Now, I'm a caver, I'm a diver. You know, if there's bones there, I'll do it. However, I am not a cave diver. I like keeping those two aspects very separate. So when I dive, like in a cenote, it's open water. I'm not trained for doing the type of cave diving like they do, where you're actually going way back in and having to use rebreathers and high-end equipment. So the most technical I've gotten is working here in Belize, where we're using nitrox, a mixed gas, to increase our downtime for diving, but we're not getting into the high-end cave diving. I leave that to other people who have a much better skill set with regards to doing that. And of course, this is what a project that Blaine and I are working on together in Mexico, in Quintana Roo, at a place called Hoyo Negro or Actunsac, where they're getting animal remains way far back in. But that's not the case of what we're doing here in Belize. We're staying just in the cenote, although other divers have gone down to 200 feet at the bottom of the cenote and have seen that it does connect with a cave system.
So Greg, I agree with you 100% on caves diving, but not cave diving. And what I wanted to ask you though, with this is, are you saying that right below you where you're working, it goes down to 200 feet?
Yes.
Okay.
Yeah, so the deepest part of this cenote in Belize goes down to 200 feet, but when you go in at 70 feet, there's a ledge with clay on it. So you can sort of see the ledge here. Here's where it drops off into the abyss of the cave. But on this ledge, there are clay deposits. So this is actually where we're recovering the bones. So we're only diving down to 70 feet to go after, or to get to the ledge where they were finding the bones when they were surveying for the archaeological material there.
Okay.
So basically, you start out, it's kind of like underwater prospecting. You just basically go down, get to the ledge, and then sort of do a very systematic moving back and forth on the ledge, keeping your eye open for bones. And you're at 70 feet, you still have a fair amount of light coming down, but a lot of times we'll also carry flashlights with us, underwater torches, in order to sort of improve what we're looking at. And of course, you've got ledges and nooks and crannies that potentially bones could be in as well. So here's just some more shots of us. These are, I'm actually in the picture here, since we had a person who was an expert on photography and underwater photography. So he was able to document that. And so Tony Rath, who is one of the people that's involved with this, gets credited for the pictures. And so you can see we're going back and forth here, checking out sites. And I don't know how many people spotted that immediately, but that's actually a bone there. So you can see you've really got to have a good search image in terms of spotting it. And of course, big material shows up a lot easier than small bones.
So Greg, is your method just to sort of fan away the surface?
Well, actually, as you can see in the upper right-hand corner, I am fanning there. And of course, when you're underwater, if it's really fine particulate material like clay, it can go up into the water column, obscure your vision, and make it hard to see what you're doing. So part of being an underwater excavator is actually learning how to fan and to move the water currents so that it's removing the sediments, the fine material away from you and not kicking it up into the water column because it takes so long to settle out. And one of the things I did when I was a graduate student at Florida, I actually worked for the state underwater archaeologist at a site called Warm Mineral Springs, and they had more equipment, including an underwater vacuum, which wasn't used for sucking up the sediments like you see a lot of the shows on TV, but was actually used to keep the water column clean of suspended mud so you could see what you were excavating and be very careful in terms of where you were putting your tools in that. But anyway, that bone that we spotted there turned out to be a complete femur of a sloth. So it was like, as far as I was concerned, the trip was already a total success. We had sloth. What more could you ask for? And when we went and checked out a second cenote that we could drive to and haul all the equipment in that, we also found bones in it as well. So we know for sure that 2 out of the 20-odd cenotes there are producing Pleistocene material. And it's very likely that as we get funding to go back down and do more exploration, we may find that a lot more of those cenotes have Pleistocene material. So there's multiple lifetimes of work here because, when you're diving, you've got a limited air supply. The deeper you go, the shorter your amount of working time. And so it's not like you can sit there for 8 hours and excavate away. You basically go down. In our case, we could be down for about 2 hours. We come back up, and then we, of course, have to blow off the nitrogen and decompress a little bit before we make our second dive later. So generally, we are doing about 2 dives a day, one morning, one in the afternoon, and the first dive generally runs about 2 hours. The second dive is about an hour and a half so that you don't exceed your nitrogen buildup in your blood and tissues and stuff like that. So one of the things you have to keep in mind is that when you're diving and you're excavating underwater, you've got to be really efficient. You can't waste time. You've got to be really focused on what you do, but you can't rush it because at the same time, you've got to be documenting, you're taking notes. And of course, the one thing you do gain is you're working in 3 dimensions. If you think about it, when you're excavating, you're sitting in a cave, you're sitting on the cave floor and you're excavating. But I can change my buoyancy, float above it, get down on top of it, and get into a lot of different positions to gain access to how I excavate that you wouldn't have in an open air site. So it's kind of a neat experience, and you have to really think 3-dimensionally when you're excavating underwater. So here we are. We've come back up with that small piece of bone that I showed you. And of course, so one of the things you can do, of course, between dives, besides writing up your field notes and that, is start identifying what you've brought up and take care of course, refilling your tanks to go back for your next dive. And so there's a lot of sort of logistical stuff that goes on between the dives that you don't normally think about. When you're sitting in a cave, if you want to take a note, you take a note. I have an underwater tablet that I carry with me that's attached to it, and so I can make quick notes. And if I carry down excavation tools or something like that, I always make sure they're tied off to me, because if I drop one over that 200-foot ledge, I'm not going down after it. So, as I said, that was a sloth bone that we found, that we had the femur there. And it turns out to be the femur of the giant sloth, the Rhemaetherium laurelardi. And, you know, we always use the term giant ground sloths. And of course, not all of them are giants. Some of them are quite small, size of a dog or so. But a Rhemaetherium was the largest. We hear about Megatherium as the giant sloth. A Rhemaetherium is actually larger. So Megatherium is the second largest of the giant sloths. And these animals are pretty well known. The skeleton in the upper left-hand corner came from Panama, collected by a Smithsonian crew. And there were 2 of these on display in the mammal hall at the Smithsonian for many years. But I think that at least one of them was taken down when the Smithsonian revamped their fossil hall. So I haven't been back since then, so we'll have to see. And of course, I always like to show this one because the animal's down on all fours, and 99% of the time when you see depictions of sloths, it's like the one in the lower right-hand corner there where they're sitting upright. So I kind of like to remind people that sloths actually walked around on all fours, and of course could get upright to feed in trees like a browser, like a Rhemaetherium was. And of course, because it's such a big animal, it made it easier to spot its bones underwater. There may be other smaller animals there, but they weren't showing up. But we did see enough bones scattered over this ledge at 70 feet. There's a lot of sloth material down there. So there's a lot of potential in this one cenote to keep us busy collecting for a long time, and not even thinking about the other ones. So there's something there for future generations to go.
So Greg, everything that you've seen so far is a Rhemaetherium?
Of the big bones, yes. Yeah. But, you know, I'm expecting other material to show up once we can more systematically work this site. This was a reconnaissance trip. Our permit from the government of Belize was to collect just a few samples to document that there was material there. And we were not set up for a full-fledged excavation. And so, for example, that femur we found is still down there. That remains to be recovered. And there's a lot of other bones down there that still are waiting to be recovered because we were only permitted for a few specimens. And this was a reconnaissance to give us enough data that we could go back for funding to do follow-up work. And Rhemaetherium has been referred to as the Panamerican Sloth. If you look at this map of North and South America, you can see all the little black dots. So its range goes from southern Brazil all the way across through northern South America. Very good record here in Central America. It's been reported. Gets into the Gulf Coast and the Atlantic coast of the United States. So of all the sloths, this has the greatest latitudinal range of any known sloth. And the fact that it goes from South America to Central America, right in North America, I think, truly lives up to being a Pan-American sloth.
Greg, is that the same species that's on exhibit at the Florida Museum of Natural History, standing on its hind legs?
No, the one at the Florida Museum is Steinobodistes, which is a Miocene sloth.
Ah, okay.
And a Rhemaetherium, the genus goes back to at least the Blancan, to the Pliocene, and it survives right up until the end of the Pleistocene. I actually think it disappeared from North America earlier than it disappeared in South America. So I think there was a range reduction through the late Pleistocene, and their youngest radiocarbon dates that we have run around 11,000 or so, but not in North America. In fact, there's only one radiocarbon dated specimen in North America, and it's about 35,000. And if you look at most of the records for the Pleistocene, they're Sangamonian. And there's very few that you could really say get into the late Wisconsin. But once you get down into Mexico, farther south, you do get later records. And then the youngest records that we have dates for are in South America. So this is one of these animals that we know it's there, but we don't really have a good chronology of its rain reduction or how it died, whether it's humans hunting it, whether it's climate change, environmental change. And we really need to have more work with good radiocarbon dates to really look at the chronology of what was going on. One of the few dates that we do have was actually off of this tooth. And this tooth was found right next to that femur. So the fact that we swam in to look at the femur and we saw the tooth looking there. And I had actually told Lisa when we were prior to the dive that if I found one thing on this dive, I wanted a Rhemaetherium tooth because we could do stable isotope work and get some paleoecological information. And we swam down, I was looking at the femur, I happened to glance over, the tooth was sitting right next to it. And it's like, I will never do this again in my career, being able to call my discovery us. So this was very exciting. And this is the tooth. We got a radiocarbon date, and you'll notice that it's from the last glacial maximum. So we're talking about the time when you've got continental ice sheets in North America, lower sea level. And so it's kind of a very interesting time to see what was going on in Belize based on what we're finding for this sloth here. And of course, the nice thing about sloth teeth is they're ever growing. They have an open root cavity. So they're like mini versions of mammoth and mastodon tusks in that they record a period of time. Obviously not as long as what you get with the tusks, but it is interesting to see that you can get different seasonality looking at these teeth because they're ever growing and they're self-sharpening. Here you can see the upper and lower teeth of Rhemaetherium are interlocking, and basically they get the vegetation in their mouths, they chop it up into little bits. And we have actually found specimens from tar pits in which the chopped up plant material is still in place, and the length of the plant material matches the difference between these loafs here. So sloths have, they're frustrating in some regards because you can't really tell the age of an individual by the stage of tooth eruption like in other animals, but they're very useful in other regards. So you gain something, you lose something working with these critters. So it turns out that there was a stable isotope program going on at the University of Illinois there in Urbana-Champaign. And so one of Lisa's students took the tooth, sectioned its length. You can see the different types of dentine. This is a little bit harder than this, which is why it's a positive relief and this gets worn down. So this is part of that self-sharpening of that tooth as it grows. And then she sampled it, its entire length, in order to look at the stable isotope record that's preserved in this tooth. And basically what we saw based on changes in the carbon isotopes and the oxygen isotopes is we could see that this sloth had basically died during a wet season, had survived a long dry season, and had come out of a previous wet season. Now, whether this represents a year or whether there was two of them, we don't have any absolute chronologies of the seasonality, but it does tell us that this part of Belize at this time during the last glacial maximum was going through alternating wet and dry seasons, and the dry season was longer than what we could see for the wet season. So it tells us something that these animals were probably going through changes in their diet, and we see that also in terms of looking at temperature averages, looking at the carbon. We can see here the change in precipitation through the year. So we're getting a lot of good paleoecological information that gives us some better understanding of what type of environments and what type of climatic changes that these animals were used to dealing with. And of course, this becomes very useful when you start getting into what caused their extinction. Was it humans? Was it climate change? This is one piece of the puzzle. We need to do this for a lot more sites over that whole range of latitude from as far north to as far south and tie it into a chronology. So this is just sort of the tip of the proverbial iceberg in terms of some of the interesting information that I think is going to come out of the study and one piece of the puzzle. And if you look at these patterns here with regards to when is the precipitation with the increase, that's running from May to November. And if you compare that with the modern data for and climate data for Belize, it starts giving us an idea of the amount of change that took place from the Pleistocene today in terms of the weather patterns that are going on. And of course, the weather, the amount of precipitation, the temperatures affecting the vegetation, these are mega herbivores. So there's going to be a secondary effect in terms of their food supply and degree of adaptability to seasonality. So when we're talking about sloths and we're thinking about their modern relatives, you know, the tree sloths, they're in tropical rainforests, very wet habitat. But here we have sloths that are obviously dealing with long dry periods and certainly not always having to be in a tropical rainforest and maybe in a dry tropical forest. And that's one of the things we always have to keep in mind, that when we're talking about the tropics, there's a whole wide variety of different environments in there. So even though you're near the equator in Brazil, you can have tropical rainforest, you can have dry thorn forest that the Brazilians call the caatinga, and then you, of course, can have open grassland savanna habitats like the Cerrado. So we have to keep in mind that this is not a homogeneous environment now. It wasn't back then. And so different sloths were probably using different types of environments in terms of the vegetation they were eating and their preferred habitat. That small bone that I brought back from the other cenote, well, here's the two halves of it here, actually was the shaft of a femur of a juvenile. So it's kind of exciting that we not are only getting adult material in these cenotes, but we're getting the juveniles as well. So tells us that we had a viable population in the area of adults and of young. And it's a very young animal. It's missing the epiphyses, or the caps on the end here. And of course, most of the time we're looking at adults, we're looking at nice complete bones. People don't pay that much attention to juvenile animals, and especially if it's the postcranial skeleton. But if you look at other sloths, Megalonyx, Nothotheriopsis, Paramylodon, these are also the shaft, and you can see the proportions and the morphology is different. So, you know, one of the things we really need to be doing is spending more attention to the youngsters and how to identify them. And as you can see, going from this to the adult, there's a real change in size and proportions of these animals and the growth. And if you didn't have these series, you might not make that association that this is a juvenile of this animal here. So this is the other exciting thing, is that we may actually, with the large sample coming out of these cenotes, be actually looking at being able to study a population of sloths from this region rather than just dealing with a single isolated find. And that's one of the things I'm looking forward to when we can go back down there. And of course, not just from Cara Blanca, but Blaine, as you and I have talked about, there's other sites in Belize that need to be explored and really build up and see what the diversity was in the Pleistocene in Belize, along with the sloths, and what other animals were living in the area with them. And I'm hoping that this will show up in our cenotes as we do more work there. And actually, as a first step in that direction, here is that shaft again, and it's got some carbonate limestone that was deposited on it, because you're in water that's very saturated with calcium carbonate. And while looking at it, that little thing showed up, and that is a tooth of a crocodile. And it's really interesting, when I was going in to make my first dive into the cenote, some of the support crew were going, "Okay, Greg, when you're in there, watch out for the crocodiles." And I'm going, yeah, give the newcomer a hard time, get him all worried about it. Well, it turns out that there are still living crocodiles in Belize. This is Crocodilus morleti. Here's its distribution. You can see it's found throughout Belize and going into Mexico. And this is not like a saltwater crocodile. This is a freshwater crocodile that actually swims up the rivers and is found in the cenotes. So when they were telling me to watch out for the crocodiles, they were actually being sincere in terms of what it was. And we have fossil crocodiles there. So there was a crocodile, fortunately, while we're live here on video, it's not live there.
Greg, is there any indication that that is associated with the bone itself? Did it come out as a bite?
No, it's actually just, it was probably a shed tooth. It just settled down and was cemented to the side of the bone by the calcium carbonate. But one of the things I'll be interested in looking for is, do we find bite marks on any of the bones in the cenote showing that crocodiles were feeding on the animals as they were in there? Because once again, you have to keep in mind, we're diving down 70 feet. When the animal was getting onto that ledge, that was probably a dry ledge. One of the other bones we recovered was part of a vertebrae here. And I could tell that's a sloth because it has this groove here in the side of this thoracic vertebrae for where the vein went off. And one of the things that's really interesting about sloth anatomy, and it's part of the fun of studying them, is they do so many weird things with their anatomy. As John White, my former professor, said, they never read Gray's Anatomy. And if you look at a sloth vertebrae, where the neural canal is almost as large as the centrum. They have a really huge neural canal, which has not only the spinal cord, but there's a very large vein that comes back. So the lower part of the body drains by going through a vein coming off through the centrum, connects with this one here, takes the blood forward, and then when you get to the anterior part of the thoracic part of the vertebral column near the heart, basically you have 2 or 3 of these where the branches come back and go back into the heart. And so it's a very distinctive feature of sloth vertebrae, and that we picked that up. So even though that was just a centrum and it didn't look like much, I could say, hey, that's another sloth bone out of the site that we got. So these were our samples, enough to document what we were finding. Now, the thing to keep in mind, as I talked about before, this is the last glacial maximum. And of course, when you've got all of this buildup as continental glaciers, you have a drop in sea level and the continental shelf gets exposed. Not a whole lot there at Cara Blanca, certainly much more from Florida, but this basically means that when you drop the sea level, because you've got a limestone platform, the water table drops. So there's no surface water. So what are these animals drinking? Well, they're basically climbing down to where the water is in these cenotes, which are providing access. And so what you're getting is animals who are somehow scrambling down into the cenote, getting onto that ledge in order to get access to water, and apparently not being able to scramble out and becoming trapped. Hence the Cenote of Doom. It's trapping them. And we see that not only at Cara Blanca, but we see this up in Mexico at Ojo Negro and other cave sites up there. And then, of course, even in Florida, where I worked with the state underwater archaeologist at Warm Mineral Springs, it was the same thing. There we had a ledge at 45 feet that we got a sloth, a saber-toothed cat, and other animal remains. So what we're really seeing is how much the water table in these areas are being affected by the continental glaciers, the drop in sea level, and how it affects the water supply. And of course, that also impacts the vegetation that the animals are eating. So you really, when you're looking at these sites, you've really got to tie it in to a broader context with regards to what's going on through time and changes in the climate, how it's impacting the vegetation, and of course, how it's impacting the mega herbivores like the sloths. So just to sort of put that in perspective, here we can see that there are lots of fluctuations over time in the water level, sea level. And of course, that means there's going to be changes in the accessibility of water. And if you have a low water table and climatically you have a drought period, then these animals are going through a lot of stress. And so you've got these alternating periods. And here's where the Cara Blanca specimen fits into the sea level curve. And you can see that as it moved even into 20,000, it would have been even more. So there may have been a time when the water table was not at 70 feet, it was lower down in that cenote. It might have been 100 feet lower. And these cenote has a steep sides, and so there's no way for the animals to get to the water. So I think that one of the things that we'll be looking at, and the stable isotopes help us tell us, is about the amount of stress that's being put on not only the sloths, but any other animals in the area in terms of availability of water and of course the vegetation in that. So we see this then that these underwater cave systems might have been open to the air. And it's very possible that some of those other cenotes at Cara Blanca, when the water table dropped, may have had easier access to getting down to the water than at the one that we were in. But in the cenote we were looking at, it's steep-sided walls except on one side where this shelf was at 70 feet that we were getting the bones off of. And once again, just the reminder that even though you're in the tropics, it's not all tropical rainforest. Here's a whole variety of different types of vegetation habitats that occur just in Belize. So extrapolate this to the rest of Central America, down into Northern South America, down in through Brazil, and you can see there's a real patchwork of vegetation that different mega herbivores might have been using. So they might have been niche partitioning, they might have been specialized for one habitat, but as that habitat changed in its distribution and expanded or contracted depending on availability of water in that was impact the populations of those animals. And so it becomes an opportunity to really start understanding the detailed paleoecology of these animals. Obviously, my bias is towards the sloths, but this would apply towards the gomphotheres, towards toxodonts, towards a whole lot of other herbivores as well. And since we're interested in the vegetation and we're interested in what they were eating and making inferences from the stable isotope and other data, part of the project that we're doing at Cara Blanca is actually collecting wood samples. And so you have, because of hurricanes and other disturbances, you get a lot of trees that are pushed down or knocked over into the cenote. And as you get deeper in the water, it becomes aerobic. The wood is preserved. And so what we're doing is we are collecting slices of these logs and being able to start looking at tree rings to look at climatic information. We're also going to be doing radiocarbon dates to provide a chronology to see if we can get some of these going farther back in time and put together this as an independent means of reconstructing what the paleoclimate was like. And of course, we're also doing that for trees on the surface to tie that into modern climatic variation. And so if nothing else, this part of the project is putting me on the cutting edge of the science we're doing. So here we are on cutting edge science in Belize. Thank you.
Greg, I think one of the really exciting parts that I'm hearing about this research is that you are getting good preservation of the tooth, at least, for radiocarbon dating. And this is something that's incredibly rare in Central America, in the tropics, that we're getting good protein out of specimens. Do you think it's because of that particular level that they're coming out of? Is it anoxic? Is it encased in clay?
All the bones are encased in the clay. Of course, the ones on the surface, the upper part of the bone is exposed to the water and potential for leaching in that. And of course, the tooth was small. It was sitting on the surface, which is why I spotted it. So when they said they were able to get a radiocarbon date out of it, I was very pleasantly surprised because based on my experience with material that we had removed from warm mineral springs and other underwater sites, they tend to be leached out. We had the same problem with a cave site in the Dominican Republic that had sloth material, that when the sea level was down, it was an open air cave, sloths were going in, they were dying. The sea level came up, it's now a submerged site, you can only get into it by diving. We tried radiocarbon dating the bone, all the collagen had been leached out.
Right.
So
I think,
it's really catch as catch can.
yeah, I think that's right. I think we have this general idea that these underwater sites like the ones in Florida and many of the ones in Central America just aren't going to preserve it. But there's so many opportunities for us to try now and really sort of change the way we look at these Central American faunas if we can get DNA out of them or if we can get good collagen out of them.
Yeah, and it doesn't have to be underwater. We have a site in Belize where we got the Shasta ground sloth, Nothotheriops, and it was a dry cave, and the bone was still leached out, and they couldn't get a radiocarbon date because there was no collagen left. So, I mean, tropical environments, because of the heat, humidity, and that, you know, you really do need to luck out in terms of having just the right, you know, the Goldilocks zone for radiocarbon dating.
All right, well, let's go to some other questions. Chris, do you have one?
Yeah, just thinking about that tooth. I mean, those things are monsters. And, you know, thinking about the fact that they're made entirely out of dentin and there's no enamel in it, so they're somewhat softer, how they have to keep growing. And so can you kind of maybe talk to the idea of how quickly do they grow? With that stable isotope data, you probably are able to kind of put some age boundaries on that and estimate the rate. But how fast do they grow?
The total length of a tooth that we've seen generally runs to about a year to a year and a half of what's there. So you know, you could sort of say, I've got a year and a half a tooth and the tooth is this long. You can sort of say, I must be getting an increment of so many millimeters per week or, you know, per month or something like that. But,
Yeah,
you know,
that's
once
looking
again, go ahead.
Yeah, that's looking like 1 to 2 centimeters a month, which is, you know, that for stable isotopes, that's fantastic.
Well, this is why I think we need more of this to be done to really do some comparison. And I've often thought that a lot of the stuff that Dan Fisher's been doing with regards to looking at proboscidean tusks and growth and what they're doing is directly applicable to looking at sloth teeth in terms of the information. And Jean Lamar, the student that did the stable isotope work, actually sampled the hard and the soft dentine to compare to see if there was more leaching in one than the other. And, you know, as it turns out, the hard dentine is kind of like enamel. It's better to sample than the soft material. And so that was one of the interesting tidbits that came out of that study as well.
Let's go over now to any questions we might have on Facebook.
Sure, we've got a whole bunch that have come in during the talk. I'm going to start with, so we've got, this is 2 questions. Mason asked, has anyone tried sifting the material for microvertebrate remains? And Jenny asked, how well do the cenotes preserve plant material?
Okay, it is my sincere hope that we do get microfauna out of these deposits, but we were not set up to collect the samples to do that when we were there because of the reconnaissance. That is something we'll do when we actually go back and do it. And of course, what we'll do is bulk collect the clays, take it to the surface, and do our screen washing outside of the cenote. So that's one of the things we'll do. We're hoping that we'll at least be able to get pollen out of the clays. So one of the things we're going to do with the clay layer is actually take cores and see if we can get a pollen sequence out of it. All of the wood that we saw was sitting on the surface. Because we didn't do any major excavation, we don't know what the plant preservation potential is down in the clays themselves. But I'm hoping for a holistic study that gives us the small animals and the plants as well as the big animals.
Excellent. I know that, I know I'll be excited to hear about microvertebrates, and I'm sure Blaine would too. But let's talk about macrovertebrates. We've got a couple of questions about a Rhemaetherium. Aaron asked, what was the niche partitioning like between a Rhemaetherium and Megatherium with regard to habitat? And Gino asked, was a Rhemaetherium gregarious? And I'll add on to that, was that also different between different sloths?
Okay, number one, if you look at the distribution of Megatherium, it is a more temperate, living in more temperate environments. You find it farther south in Argentina. You find it in Bolivia. You'll actually find it high up in the Andes. So Megatherium is living more on the pampas and more open country, sort of a savanna habitat with scattered trees. And its latitudinal range is much farther south. It does not get into North America. It was basically stopped by the rainforest environment that you find in northern South America. Whereas a Rhemaetherium is definitely associated with the more tropical latitudes from, like I said, southern Brazil, and then subtropical. The northernmost record of a Rhemaetherium is in New Jersey. And it probably was up there during the Sangamonian interglacial and not during a glacial. So you actually don't find any areas where the two genera co-occur. They seem to be very different, and it seems to be separated by both latitude and elevation.
Ascanio, did you have any questions you wanted to ask? He's joining us from Venezuela today.
Here.
There you go. Oh, you're muted.
Here. Yeah, you hear me?
We hear you.
Yeah, I wonder how many caves there are in Belize in terms of potential of looking for fossil in the future in there, and if there are any speleological society doing the maps in the area that will be help in the future projects in there.
I'll defer to Blaine on that because he's been down there checking out caves. I've only been in the cenote.
Oh,
So both in Belize and in the Yucatan of Mexico, there are just so many caves, and a lot of them are underwater because of sea level change. So there's that aspect of it, that we're still discovering these caves and these underwater caves as divers are starting to discover them. So there's just a lot of them. But as you move more west, then you get more into these drier caves as well. And again, there are a lot of them. And yes, there are speleological groups that are looking and discovering and mapping them. But a lot more needs to be done. And the paleontology is really just starting. So come and join us.
yeah, sure.
To get back to the second part, whereas Rhemaetherium gregarious, I'm Greg, so I think they're gregarious. But actually, we do have a number of sites where multiple individuals of Rhemaetherium have been found. Some tar pit sites in Ecuador and Peru. There was a site in Florida that's been found that had lots of them, and in Brazil. So they do appear to have been maybe large family groups, maybe not huge herds, but probably some type of extended family group. So the fact that we find multiple individuals together in single sites suggests that there was some type of quasi-herd or gregarious behavior.
Okay, and on a similar note, over on Twitter, our friend Dr. Alton Dooley sent us a question. He says, Greg, how sure are you that the juvenile femur is a Rhemaetherium and not some other taxon?
We have other, to confirm that, I spent some time at the Florida Museum of Natural History. They had a sinkhole deposit from the Hale area, Hale quarries, and they had both adults and juveniles of a Rhemaetherium. And I actually was able to see some other Rhemaetherium juvenile specimens that match up with the shape, morphology, and proportions of the one that we got from Belize.
Gotcha. We've got another question here from Barbara, who asks, why is it that you can tell the age of a fossil better from a tooth than from other kinds of bones?
Actually, that's simply because most people focus on the teeth. One of the things that I've been trying to work out is looking at the juveniles, is look at the stages of epiphyseal fusion, when the ends, at what stage, and the order in which they fuse onto the shaft and formally attach. And that's one of the advantages of Rhemaetherium, since you do get these large samples, that you can get juveniles as well as adults to at least work out the relative sequence. I'm not sure we're going to be to the stage where I can tell you this was a 3-year-old, because we don't really have the equivalent of African elephant ears like we use for mammoths and mastodons. So the best I might come up with eventually is a relative stages of growth that allow us to make some reasonable groupings of really young juveniles, adults, really old adults. But we don't really have anything that gives us an absolute age that we can say this was 1.5, this was 2, this was 20 years. That's kind of a holy grail for me in terms of, another Indiana Jones reference. But basically, there's a lot more that needs to be done. And one of the challenges with sloths is they don't always look or do things quite the same as other mammals.
All right, and then I'd like to ask you a question inspired by a couple of comments we've gotten throughout your talk. I saw a number of people commenting, looking at you describing the process of diving through these caves and just saying, nope, terrifying, would not do that. And then I saw at least one person who said, that seems awesome, sign me up. So my question is, how difficult is it when you're trying to do work in a place like this, to find people who are willing and or qualified to participate in this kind of excavation?
The person who is our dive master, Chip Peterson, is a certified cave diver. So he's one of those people that says, sign me up. He likes the adventure of going down into submerged caves and that. So basically, you just have to look around and find people who have that level of adventure and have taken the time and effort to get the training. You start out as a regular open water diver, which is what I have, and then you can take specialized training courses that teach you all the safety and requirements that's needed to do cave diving safely. The people that Blaine and I work with on Ojo Negro in Mexico are trained like that. Blaine and I are happy with the open water. We turn over the technical work of going into caves to them. So cave diving is phenomenally dangerous, and you really have to be highly trained to understand all the ways that you can do yourself in if you're not careful, and what are all the proper precautions to take. And then you can do it. But in terms of the cenote, since we have open air above us, if anything happens, we can go straight up and we know we're going to hit oxygen again. That's not the same as a submerged cave where you bump against the roof and there's still water there and no air pocket. So getting people who are open water divers, then it's just a matter of teaching them the underwater excavation technique. But once you get into cave diving, that is orders of magnitude more challenging.
And the sites that are in these underwater caves, they are basically reported to us by these cave divers. And then it's those cave divers that we end up working with and want to help and work with these projects. So we're never really looking for another cave diver to help us, because we're working with the one that found it, usually in the first place. In these open cenotes, though, there's always a risk and there's a level of danger. But once you start going back into these caves, it exponentially goes up. And it's not just more qualifications, it's a lot more time and money to get those qualifications. So it's something that you have to think of. I'll be doing that in years from now, potentially. It's not something you rush into.
Yeah. Now, Ascanio has actually done diving for fossils in Venezuela, but he was actually looking for fossil shark material from a site off on some islands off the coast, right, Ascanio? Tell them a little bit about what you did.
Yeah, we found a turtle and a pelican into the water. That was my first time diving, so that was really, really dangerous and crazy. but I did it. I survived, and we yeah, we studied that material. But there are many potential caves in here. I mean, in Venezuela for diving for looking for fossils. And actually, we have one in which we collect some sloth material, and we have the plan to back when we found some money or something. But it's not so dangerous. It's just like a siphon into the cave, but the other side is dry and have a many fossils in one corner. But we suspect that we have a chimney in the roof of the cave in the other side, in somewhere in the jungle. So we expected to find the other entry to get into that cave. That will be very interesting because it's like a trap pit. But we wonder if a carnivore house or is actually a trap pit in there. But it's not like the river accumulate the material into the cave, but probably it's another taphonomy in there. So it will be very interesting because all the area is, full of holes, like trap pits in the area. It's a enormous karstic system in the Perihar Range. Is the western Venezuela. So we have another area in which we found some other sloth in Falcon State, close to the coast, where we actually have a cone of bone into the cave unexplored. So for that reason, I asked you, how potential is Belize in terms of fossil and caves?
Hay mucho.
We'll have to get you on the show sometime, Ascanio, to talk about those sites.
Please. Yeah, please.
All right, everybody. Well, we are just about at the end of our program. So, Blaine, if there's anything else you'd like to say or ask.
No, just thank you again, Greg, for an excellent presentation. And thank you, Ascanio, for joining us as well.
Oh, and thanks to everyone for watching and asking questions. And we'll see you next week, I suppose.
Yep, next week.
Bye, everybody.
Bye-bye.
Take care.